It's a pleasure to welcome Dr. Brent Harris, who is a tenured faculty member of Georgetown University with dual appointments in neurology and pathology. As a neuropathologist and physician scientist, Dr. Harris has clinical, research, and teaching interests in neurological and oncological diseases. He has active collaborations and research programs in his own lab in the areas of neurodegeneration and in CNS neoplasia. His primary interest is in understanding how mechanisms of neuroinflammation and glial neuronal communication influence the pathophysiology of neurological diseases. In addition to investigating disease processes, he also seeks to uncover targets for pharmacological intervention. I will now turn it over to Dr. Harris, who will be talking to us about ALS.
Good morning, gang. Okay, let me get my screen going here. 1 second. Okay, hopefully we are in good shape. Thank you for that introduction, and thank you to my co-committee folks from the Education Committee of the AANP, Dr. Oviera and Dr. Martinez-Laje. Really appreciate this invitation, and I look forward to chatting with everybody today during the Q&A. I'm going to give you guys an update on ALS, and it's going to have my own flavor. I think this series has been really awesome in terms of covering a wide variety of different topics, and ALS is one that is a primary in my interest. So with that said, I am going to, let's get the pointer going here. And I welcome anybody who's live. I welcome you guys who are maybe gonna hear this in a recording later on. My contact information is right below. If you ever have any questions about the things that we're doing about ALS in general, feel free to contact me. I also want to thank AOE for the awesome coordination of these talks. They do a really good job behind the scene, and really appreciate that coordination. So I really don't have any conflicts for this talk. My funding sources are listed above. I do have a Neuropath Consult LLC that I do for some of my medical legal work, but I'm not going to be talking about that today. My specific learning objectives for this talk are to define the evidence that ALS and some frontotemporal lobar degeneration diseases lie on a disease spectrum, that I'm going to describe some of the histopathological findings that define ALS and FTLD, motor neuron disease, and I also want you to be able to describe some of the genetic alterations found in familial and sporadic forms of these diseases. So here's kind of the breakdown of topics. We're going to do some very basic stuff at the beginning about ALS and FTLD. And then as an illustration of some of the history of this over the last 30 years, I'm going to talk about some of the collaborative stuff that I've been doing with multiple different laboratories. And if we have time at the end, I'll talk about some of the biobanking things that we are doing right now. So some basic facts about amyotrophic lateral sclerosis. It's a neurodegenerative disease causing progressive paralysis, leading to death in most folks, though not everybody, in 2 to 5 years. It's really highly variable in terms of the early symptoms and patterns of progression. And that makes it very tough for neurologists to make the early diagnosis. And many patients don't really come to clinical diagnosis because the symptoms are very subtle to them as well. Most cases of ALS are sporadic, or means we really don't know the reason that causes them, with probably 20–25% now being recognized as familial. We'll talk a lot about this difference. I'm not going to talk a lot about the treatment of ALS, though there are 2 FDA-approved medications. Riluzole has been around for many years, and Radicava has just recently been approved in the last few years. But these guys both offer a minimal therapeutic benefit. So the name ALS, it actually has other names, as you might expect. It's often called motor neuron disease in many parts of the world. Really, Charcot, the father of neurology, was among the first to do a lot of the descriptive work describing ALS, and so in France they still call it Charcot's disease in some places. And then in the US, we also call it Lou Gehrig's disease, based after the famous baseball player of the early 20th century who developed this late in his career. And it's kind of ironic because he was known as the Iron Man. He played on and off the field, regardless of injuries and sickness, for many years, back-to-back games, and held the record for a very, very long time of the most consecutive games played. But during my childhood time, that record was broken by one of my local favorite baseball players, Cal Ripken. So the very quick review of Neuroanatomy 101, because these are the cells and pathways that we're going to be talking about today. So the motor pathway originates in the frontal lobe, the motor cortex, with the homunculus that you don't see here, but with differential representation along the motor cortex of the right and left sides. The corticospinal neurons are large layer 5 neurons that send long projections either down the pyramids, the corticospinal tracts, or to some of the cranial nerve nuclei. Remember that the cranial nerves, some of the cranial nerves are lower motor neurons. And in the lower medulla, most of the fibers decussate to the opposite side and then travel down the lateral corticospinal tracts, or some of these anterior corticospinal tracts. They synapse either directly or indirectly, sometimes with lower motor neurons that send their very long projection out to the muscle group that they're going to innervate. So this is a kind of a busy slide, a lot going on here, but it's a nice summary slide of the wide variety of different motor neuron type diseases that you can see out there. So classic ALS is usually broken down clinically into bulbar onset or spinal onset. Traditionally, bulbar onset portends a more rapid decline, but not always. And some people present with both findings often early on. The other specific subtypes, these are all clinical specific subtypes, and we don't necessarily recognize them as pathologists very differently. We do recognize, though, some of these subtypes that are really only upper motor neuron or lower motor neuron. And the folks that unfortunately have predominantly lower motor neuron disease don't do great, because the lower motor neurons are innervating the muscles of breathing. Folks with primarily upper motor neuron disease can live a long life with some debility, but it doesn't seem to necessarily affect the lower motor neuron. To make the diagnosis of ALS, you need to have both upper motor neuron signs and lower motor neuron signs. Those are the El-Escorial criteria. A muscle biopsy can come into play, and I'm going to show you some images of muscle biopsies, but a muscle biopsy in and of itself cannot make the diagnosis of ALS. So as pathologists, we do come across folks that do get biopsy, because there is a differential of ALS and various different inflammatory myopathies or adult onset dystrophies. And so you will come across that. Now, some of you may not have known, though, that if folks live long enough with ALS, they can have cognitive features as well. And I know that when I was in medical school back in the 90s, we were taught that with ALS, you know, it's a disease just of motor neurons, and people are left with completely normal cognitive abilities, which made it in some ways a more devastating and terrible disease. Folks that do have careful cognitive testing, though, with ALS, you often can find some subtle and sometimes not so subtle cognitive problems. And many of you will recognize that some of these features of aphasia, semantic dementia, progressive aphasia, and behavioral variant or primary progressive aphasia are features often of frontotemporal lobar degenerations. And we now know that ALS and FTLDs, some FTLDs, are on a spectrum of disease, both in terms of clinical signs and symptoms and pathology and protein aggregates. So how does the name, where did the name ALS come from? So in fact, I don't even know who coined the term ALS. If anybody knows, put it in the chat and we'll talk about it at the end. The first name, amyotrophic, is a little bit of a misnomer. I think it probably should have been myoatrophic. So if you go to the Latin, this means no muscle growth. And it's not that the muscle isn't growing, it's that it is shrinking. And so myofibers, if you look at an H&E stained cross-section of a muscle group that's affected in ALS, you'll see this grouped atrophy of fibers, maybe a little bit of commensurate hypertrophy next to it. You don't see inflammation, and you don't necessarily see these guys degenerating. So the myofibers shrink and they get smaller and smaller, and that's because they're being de-innervated. The other things you can see with all the special stains that we still do are groupings, fiber type groupings. So while some fibers are being de-innervated, they may be re-innervated, and they can change from a type 1 to a type 2 or vice versa. And you find groupings of type 2 fibers on a ATPase stain or on a myosin fast, myosin slow combination immunohistochemistry, and groups of type 1 fibers. On some of the other stains that we do, like NADH, you might see target fibers. Esterase becomes activated, and you can highlight these angulated small fibers that are atrophied and show more esterase than the larger fibers. So those are kind of the three hallmarks, but you don't want to make the diagnosis of ALS if you see those, because you can see those same things with radiculopathies and motor neuron damage, a variety of other things. But you can say that there's denervation, and in the right clinical context, that will let the clinical team know that it probably is ALS. The other part of ALS is lateral sclerosis. I already mentioned that the lateral corticospinal tracts get damaged, and the virtual slide that you were provided in the invitation is from these stain slides that are highlighted with a myelin stain. So it's not that these are demyelinating, it's just that the loss of the corticospinal tracts here, you lose both the axons and the myelin, and you can see that loss here, maybe even a little bit of loss here in the anterior spinal tracts. When you look a little bit closer at the cortex of a patient with ALS, and this is a patient from our target ALS postmortem core that I'm going to talk more about at the end, you see that in that motor strip, you'll get an activation of microglia and astrocytes. So there's a local neuroinflammation, whether that's a primary cause or a reaction to the dying cells, or some combination, we really don't know. Right next door in the sensory cortex of the same patient, you really don't see those changes. You see nice tame appearing microglia and astrocytes. The other change that we look for in diagnostically to make our diagnosis of ALS at autopsy is the presence of abnormally located and aggregated TDP43. And what's fascinated me about the TDP43 aggregates are that they're not just in neurons. I think this is in the anterior horn of a spinal cord from a patient with ALS. You see the aggregates in the motor neurons, but you also can find them in glial cells, astrocytes and oligos. And it's not every cell. So normally TDP43 is localized primarily in the nucleus. So if you do an immunohistochemical stain, you'll see TDP43 in the nuclei to varying degrees, kind of throughout the body, but especially the brain. But in certain neurodegenerative diseases, you see that it mislocalizes. You don't see it in the nucleus here or here into the cytoplasm as aggregates. And this is true in all forms of sporadic ALS and in most forms of familial ALS. It's also a phenomena of other neurodegenerative diseases, especially FTLD, some forms of FTLDs, but you can even see it in Parkinson's and Huntington's and Alzheimer's disease, less commonly than FTLD and ALS. I really like this slide. This shows the genetic timeline of really the heyday of genetics for ALS, which started back when I was in medical school. And Gurney's group found or described the first family with hotspot mutations in the gene superoxide dismutase 1. And that was a phenomenal finding that was led up within another year or so of developing transgenic mice with that human stereotypic mutation, and the mouse got very similar disease to ALS. So during this time period of about 10 or 15 years, SOD1 work was really the predominant research going on. And then a few other genes were discovered during that time period, but now it's really taken off in the last 10 or 15 years, such that the next big milestone, I would say, is the TDP43 description in the late 2007, I think it came out. And the finding that abnormal protein aggregates were not just in families with TDP43 mutations, but even people that didn't have TDP43 mutations, sporadic folks. And that kind of unified the ability to do some different types of research, I would say, that is still ongoing. In the last 10 or 11 years, the big finding with C9orf72 abnormalities that I'm going to go over briefly is now taken center stage, along with other proteins that have come up in to a lesser degree. But we now know that C9orf72 is the most prevalent familial ALS genetic alteration, but also present in some forms of FTLD. Along the top here, you sort of see the pathogenesis, pathophysiology areas that were researched over the last 30 years or so, with protein aggregation being recognized early on. The SOD1 mutations don't cause a loss of function necessarily, but their altered structure causes them to get stickier and to aggregate atypically. And so we think of it primarily as a gain of function. There may be some loss of function, but it's primarily a gain of function, because if you get rid of this gene completely, at least animals do okay. The early 90s also had lots of great research on glutamate excitotoxicity, some pioneering work from Jeff Rothstein's group, and then really the mitochondrial dysfunction, axonal transport defects were recognized. If you think about it, those upper and lower motor neurons are unique in your body as probably among the biggest cells in your body. And you have those cells for your entire life. So we've had to come up with some exquisite ways of maintaining the integrity of those cells and moving things along their pathways. And if that gets disrupted, even a little bit, slowly over months and years, those cells can be damaged and die. Oxidative stress, ER stress, and RNA metabolism kind of led the pathway, or led the pathogenesis pathways then, and then proteasome involvement. And actually it was recognized that proteasomes were screwed up way back here. So now we're sort of coming back full circle again. So let's talk about C9orf72 for a minute. So two groups, one from Europe, Rademacher's, and one from the US, led by Brian Traynor. both came out with back-to-back papers and really discovered the genetic changes associated with C9orf72. This is a little more up-to-date table of genes associated with ALS, and while it's hard to see probably on your screen, you probably can see it okay. You'll get a copy of these PDFs, and you're welcome. This is from a recent Nature review. And really, as I mentioned, C9orf72 is the most prevalent. And you even see abnormalities in so-called sporadic ALS. So, the genetics of familial and sporadic ALS are very complicated, and there are overlaps. ALS, like most neurodegenerative diseases, we think has a genetic and environmental component in most cases, and it's really difficult. And this one was probably not recognized early on, partly because people sometimes present with ALS, but their relatives may present with a completely different neurodegenerative disease, or in this case, we're finding related neurodegenerative diseases. So maybe it wasn't recognized in the family per se. SOD1 is the next most common, TDP43, another RNA metabolism or binding protein. FUS is important in some cases, and you'll see the list going down here. So this is another way of looking at. So we still don't know the cause of the majority of ALS cases, but we are actively, we being the community, are actively looking at many different families and genes. And if you break it down here, the familial genes, C9orf72 is the most important. SOD1 being probably the next, and then you've got all of these other guys that are broken down here that you can see on the side. So complex genetics. So how does C9orf72 cause the disease? We don't know exactly, but the prevailing thoughts are that one or more of these RNA processing processes gets screwed up because of these hexanucleotide repeats. So of different repeats in other diseases like Huntington's and cerebellar ataxias. There seems to be and those are trinucleotide, but this is a hexanucleotide repeat that's found in intron 1 of this gene. And the thought is that could reduce expression through impaired transcription. It could alter the RNA structure and cause sequestration of binding proteins, or it could create abnormal peptides that are toxic to cells. And so this combination, I think right now, we think they're probably both gain of function and loss of function aspects to this gene and protein product. There's an active effort to make C9orf72 animals by a bunch of different groups, and they have been successful in creating these. We normally have 20 or so copies or repeats of that hexanucleotide, but when you get into the dozens and hundreds and more, that's when you start having the problem. So they had to use BACs, artificial chromosomes, because of the size of the gene, and these animals maybe are a little bit better in terms of representing FTLDs right now, because they have some behavioral changes. Their motor changes are not great, or not. The loss of motor function is not great with them, and so there's still a fair amount of effort to try to get ones that really replicate ALS a little bit better. So we now know that ALS is on a spectrum of disease with FTLDs. And you have some people who have signs and symptoms and probably pathology of both FTLD and motor neuron disease, or ALS. You have some people that really don't show any cognitive changes and have more pure ALS. And then you have people that run the full spectrum. And as I said, the genes and pathology and phenotypes are very complicated. And so the classic ALS, we know that there's TDP43 aggregates, but there are all of these other associated genes that, you know, we found abnormalities in, even in sporadic folks. We know that there are people that have changes in SOD, even in classic ALS. So you've got all of these different forms of FTDs or FTLDs and different forms of ALS. And then you've got these different aggregates, and they could be tau, they could be TDP43, they could be, you know, these dipeptide repeats. So everybody is fairly unique, and for any neurologists who might be listening to this, you know that each person who gets these diseases is pretty unique in their early manifestations and how they progress. So it's kind of important to do this phenotype genotype characterization, because we think that there are folks that do have long-term survival, and there are some folks, sadly, that, you know, have very short-term survival within a year or two of their diagnosis. So for families and for those individuals, it is important for them to know what their progression is going to be. And there may be differential treatments that you want to try depending on that. And I think there are clinical trials that have emerged to try to address this and try to understand the different forms to better figure out what treatments to use. So the ideas about the pathogenesis have spanned so many different cell types and so many different biochemical processes over the last 30 years. And if you look hard enough, you can find abnormalities in glutamate excitotoxicity, and you can find abnormalities in proton pumps. We know that there are aggregates, an accumulation of neurofilaments or TDP43 aggregates. And so I would say early on, we really took a very neurocentric approach to looking for abnormalities, because these were the endpoints of what cells were degenerating. But I think that during that time period too, glial biologists also started saying, well, wait a minute, you know, the neurons don't live in exclusivity, and glial cells have a very important role in maintaining neurons and in the normal communication of neurons. And maybe it's the glial cells or some combination. So I really got interested because I did my postdoc with the late Ben Barris, who I think was really one of the greatest glial biologists that we've had in the last half century. And Ben was really a great mentor in terms of saying, you know, ask the big questions, think about things holistically. And so we focused on both neurons and glial cells and how they interacted with each other. And here's a representation of a transgenic animal that has a yellow fluorescent protein in the corticospinal tract. So you can see the beautiful dendrites that go out in the axon that's going down. And they, you know, they live in this layer 5. This is a mouse and that's been transgenically created, but they don't live in exclusivity. You know, they have all of these other cells that live around them, other neurons, other glial cells, blood vessels that form the neighborhood. And Don Cleveland, about 15 years ago, and his team really pushed hard this idea of problems in the neighborhood, and that neurons were not the only cells involved. So you can break this down. So I'm, you know, in this next grouping of talks, I'm going to talk about some of the research I've done over the years to look at different areas of these pathogenesis, just as an example. And we really got our first start by looking at motor neurons that could be cultured. If you can simplify things, you sometimes can ask questions that then you can work backwards. And you can grow motor neurons in a culture dish. In exclusivity, they don't form synapses, but if you throw various different secretions from astrocytes back on, they actually will make synapses. We got really interested in mitochondrial dysfunction, and I think many people still think many neurodegenerative diseases are due to mitochondrial dysfunction. And the combination of inflammatory cytokines like TNF, so you can throw various different cytokines on these cell cultures and watch mitochondria. So these are mitochondria that have been labeled with a mito tracker, and normally mitochondria kind of live both in the cell body, but they also are out here in the processes. They've got to be making all that ATP to keep things moving along the processes. But if you put TNF alpha on the cultures, and these are purified cultures, the mitochondria sort of slow down. And those that are there often are kind of moving back in towards the cell body. So that means they're not out there to make the ATP needed to do all of the functions. And so I had a poor grad student that had to count mitochondria moving across, these artificial processes, either backwards or forwards. And we did see that they significantly move back in. And that's sort of one of the things that happens with apoptotic cells. The mitochondria tend to go back in. We also studied these SOD1 animals and looked at, in this case, a transgenic rat that came out in the early 2000s. And these guys, over a very stereotypical period of time, start getting paralysis. Often it's hind leg paralysis, and that progresses. And so you can then use them to look at different time points in disease, and you can look at both astrocytic activity or microglial activity. Microglia are evenly spread in the gray and white matter in wild type rats in their spinal cord. And then pre-symptomatic, before these animals get any kind of motor deficit, you start seeing aggregations of both astrocytes and microglia in the anterior horns, where those anterior horn cell bodies are living. And then by end stage, you really see a really robust microglial response. We also started studying TDP43 a number of years ago. So this is a DNA RNA binding protein with lots of different sites of interactions with other proteins and with various different DNA and RNA sequences. And it can shuttle back and forth from the cytoplasm to the nucleus. But as I showed you earlier, you get these aggregates in both glia and neurons within the cytoplasm. Here are some more examples. It gets the fragments are often truncated C-terminal fragments that become hyperphosphorylated. And as I said, they really they aren't found where they're supposed to. So maybe there's some loss of function in their normal binding function, and then they make these aggregates that are sometimes taking up the entire cell. You can create what are called nanobodies against various different proteins, and this is an active area in a lot of neurodegenerative research. So a nanobody is really just the variable heavy chain portion that shows the variability and has recognition for specific antigens. And I've worked together with a chemist at Arizona State University named Mike Sirks, who's created these nanobodies from proteins that we've aggregated from our human ALS bank cases, and made various different TDP43 nanobodies that potentially could be used both for diagnostics, but also because they can cross the blood-brain barrier, potentially for therapeutics. A new collaboration that we started just about 5 years ago was with an investigator at the National Cancer Institute. Never thought that I would, get a call from this particular investigator who, is well known for his research in P53. And he called me one day and he said, Brent, I've got a very bright graduate student in a Oxford NIH program, and she wants to study P53 in neurodegenerative diseases. I was like, wow, P53, I mean, I know that as a pathologist from cancer. And he said, well, there's some thought that there may be a role in neurodegeneration. And so, yeah, he actually did call me Brent because he knew me, because it's my dad. So how cool is that? I'm getting to research with my dad for the last few years. So Kurt Harris has been a leading pioneer in P53 biology in many different cancer cell types. But he said, Brent, I know nothing about neurodegenerative diseases, so you got to help me mentor this student, and it'll be a chance for us to collaborate together. So now Dr. Kashmir Turnquist, and she may even have her MD right now. She did her PhD and then went back to Oxford to do an MD, really led a lot of this work, and it was super fun to be able to work with her and my dad and folks in his laboratory. And so the Harris Lab at Georgetown and the Harris Lab at NCI, which is just down the road from us, about 10 miles, began studying the role of P53 isoforms in brain aging, senescence, and neurodegeneration about 5 years ago. So what is senescence? Senescence is biological aging, gradual deterioration of function, loss of cells' power to divide and grow, and unfortunately, it is inevitable in mammalians. But it can be accelerated with various different things like stress and disease and genetic mutations. And there are some diseases like progeria and Down syndrome where senescence is accelerated. UV irradiation and things that we do in terms of radiating folks and chemotherapy can also accelerate senescence. And there may be drugs and other methods to delay or slow down senescence. So there's a lot of science going on in this, including within neurodegenerative diseases. So we decided to look at the various different P53 isoforms. So this is a gene that is the master regulator of proliferation. And we know that is a tumor suppressor gene that gets altered in many different types of cancer, but it controls normal proliferation as well. And while the motor neurons are not proliferating in the brain and the spinal cord, the glial cells are very slowly. And so our kind of central hypothesis is that maybe the glial cells in some folks are going into a premature senescence. And so the two isoforms that we're studying are the delta 133, let's get the laser pointer going here, P53 alpha isoform, and the P53 beta isoform. And these are kind of two sides of the seesaw. Delta 133 is down-regulated in cells that are going through replicative senescence, and it inhibits full-length P53. And it's degraded via selective autophagy. As opposed to P53 beta gets upregulated during replicative senescence, and it cooperates with full-length P53, and it's regulated by RNA splicing. So these are sort of flip sides of a coin. And I'm not going to go through all of the data in the next few slides just because of time. But if you're interested, we wrote our first paper together, the first paper I ever wrote with my dad, which was really fun in 2016, led by Kashmir. And we took both AD brains and ALS brains and looked at some stereotypic markers of senescence that are well known to the cancer community. So P16, nitric oxide synthase 2, and gamma H2AX. And we found that in AD brains and in ALS brains, predominantly astrocytes are labeling with these markers. And we did dual labeling with GFAP to confirm that. And we found that in the tissues as well, that there's upregulation of these at both the protein level and the RNA level. Here are the protein westerns for P53, the large fragment of P53, which is upregulated by itself in AD and ALS, but then the P53 beta is upregulated, and the delta 133 is relatively downregulated. We quantified that, and then we decided to go to cell culture. These different isoforms are not found in rodents, and so you really can only look at them in humans. So we had to go to human cell culture, and we started looking at primary human astrocytes. And these cells, primary astrocytes, will grow in culture, but by the time they get to around P10 of passage, they start developing a phenotype that is called the senescence-associated secretory phenotype. And they will express beta-gal, which is a marker of senescence. In culture, they will express similar to the older age brains that we looked at, or in rather the neurodegenerative cases, upregulation of P53, downregulation of the delta, and upregulation of the P53 beta. Next, we wanted to do co-cultures to see whether or not these astrocytes that are going into senescence are detrimental to neurons. And again, we had to go to human neurons, which are not easy to culture, but you can create iPSCs, induced pluripotent stem cells, to differentiate into neurons. And then you can do neuronal and glial co-cultures. And I'm not going to go through this data other to say that if you could increase neuronal death when you cultured with astrocytes that had the delta 133 knockdown by siRNA, or the upregulation of P53 beta. And on the flip side, you could overexpress cultures with delta 133, and actually that could rescue the phenotype. Either of the astrocytes by themselves, or together with the neurons. So our model, our fairly simplistic model, is that young astrocytes are expressing more delta 133. Older astrocytes are expressing more of the P53 beta. These guys produce more cytotoxic cytokines and inflammatory cytokines that are not as receptive to keeping neurons healthy and happy, and they stop proliferating. So if they're not there, then they're not going to be making all of the normal growth factors and doing all of the things that astrocytes do to help keep neurons healthy. So now I think Ben would be really happy of me. This is an astrocentric view of the world, where the problem is really more with the astrocytes than these little tiny neurons that do something. We actually know all of these cells are important, whether it's an astrocyte or a microglial cell or a neuron. But all of these things, aging can lead to DNA damage, inflammation, things that we do as clinicians, that can lead to neurodegeneration and release of various different toxic substances into the area. And that there may be things that we can actually do to treat folks.
Sorry to interrupt, Dr. Harris. There's just 15 minutes left.
Okay, last few minutes, I'm just going to talk about some of my inspirations of becoming a brain banker and how we're using brain banking as a way to help the community in general who are interested in doing ALS research. So my inspiration came when I was a kid, when this movie came out. And if any of you in this call or who are listening to this have not seen this movie, you are culturally stunted, and you're required to go see it. I think it's still on Netflix. Classic brain banking in that movie. So we use in our ALS consortium really classic techniques of brain banking, and this is a wonderful paper from one of our colleagues, Jean-Paul Vancetel, who leads the Columbia banking effort for one of our cores. And Jean-Paul wrote this wonderful paper that shows kind of the principles of banking in general, where we're looking at multiple different areas in a very controlled way. And in ALS, obviously, we're looking at motor cortex in all of the motor strip. We're looking at the brain stem for changes, and we're looking at the spinal cord for changes. We then are storing tissues in a fixed state, in blocks, frozen. And with these frozen tissues, you can do all kinds of omics. And so we've collaborated, our Our ALS bank has collaborated with the New York Genome Center to do RNA-seq and advanced RNA studies. And now we're starting to do more spatial type omics, where you can take slides that have been fixed, formalin fixed, paraffin embedded, and you can look at protein and RNA in very specific areas. So it's now requiring neuropathologists to work very closely together with molecular biologists to come up with data. So we have a resource that's supported by a foundation called Target ALS, and our overall goals are to create various different repositories, both of tissues, postmortem tissues, and pre-mortem biofluids, do whole genome sequencing, RNA-seq, and we are looking at ALS, healthy controls, and some other disease controls. And we give out this tissue free of intellectual property, free to academic partners, and a very small fee to industry partners, where we also think a lot of good science is happening that is going to help out folks with this terrible disease. Yeah, I'm going to skip over that. I'm going to skip over that. Last two slides just showing a recent study using some of this data by Molly Hamill's group at Cold Spring Harbor, where she took 150 of the cases and the RNA-seq data and did unbiased machine learning to put them into different categories. And from this, she uncovered two categories that we expected. An inflammatory category that had activated microglia and other inflammatory markers, and an oxidative stress, ER stress category. The one that was unique, though, we really didn't expect. And this category was one that had abnormalities in retrotransposons. So these are things that are involved, right, with viral incorporation and viral disease. So now she's following up on that and looking at the interaction of these retrotransposons with TDP43. So this kind of learning, which goes from general banking to then very specific kind of new discoveries, is where we hope the field is going. So I talked about ALS and FTD sitting on a spectrum, having a characterization of variable clinical, genetic, and cellular changes. Talked a little bit about the differences between familial and sporadic forms, and the really important aspect of collaboration for these very complex diseases. So I acknowledge the families and patients that volunteer for clinical trials in ALS and biobanking. I acknowledge folks that I collaborate with and from my own lab. And I thank you guys for your attention. And we'll open it up now for questions. And I think we're going to do questions, and I'm going to stop sharing, I think, so that I can see the chat.
Thank you,
questions?
Brent. That was great. Yeah, I copied a little bit of information on the origin on the term that James Mandel provided the
awesome.
link for.
Thanks, James.
And then, yeah, you may submit any questions you have via the chat box, or you can even unmute to ask a question if you have any for Dr. Harris. I haven't seen any questions coming through yet.
You guys all know the causes of ALS, huh?
Hey, Brent, this is Jim Mandel. Can you hear me?
Yeah, hey, Jim.
I have a question, a practical question. When looking for signs of tract degeneration, what are your favorite immunohistochemical markers? I know that you showed a nice case with the LFB, but that's a sort of a very end stage severe example. What about more subtle tract degeneration?
Yeah, so you can look at for tract degeneration both in the brainstem and in the spinal cord with like a CD68. So as those corticospinal tracts are degenerating, you're going to activate microglia to try to eat up the debris. And I've found that microglial markers are probably the most sensitive, because not every case has abundant upper motor neuron disease. And so sometimes it's a little bit subtle to see with the myelin stains. But I do both microglial and GFAP markers. Both are usually found in the tracks, as well as either LFB PAS or LFB H&E stains.
Thanks.
Okay, a couple of questions are rolling in. Did you use the routine P16 IHC for that cell path? Would you use, or did you use, a different clone optimized for postmortem tissues? We tried a few different clones for P16. I would have to go back to the paper to see which one we ended up on, but it worked absolutely fine in both the cell cultures, but also in the paraffin embedded tissues. Viral infection can be the reason for neurodegenerative diseases. Wow, that's a great question, Mohammed. And one that probably has certainly predated my birth, when viruses were thought to be the causes of CJD. Viral infection in our most recent trend, SARS-CoV-2, certainly can have effects in the brain. And whether it is the viruses that are causing direct problems in any particular cell to cause the neurodegeneration, or if they're causing a neuroinflammation that has secondary effects in the local neighborhood, I think is completely up in the air and really exciting areas to research. So I encourage everybody to keep an open mind, but sadly, early viral infections can have long-standing changes later on, as we learned in the first pandemic with post-encephalitic Parkinson's disease. What is your standard workup for an ALS case to identify potential familial cases? Yeah, so there are companies that have arisen that can do panels to look for the most popular ALS changes. If your site is a member of the NEALS group, the New England ALS Association, at one point, they were allowing you to send for a panel of like 20 common genes for free. And so that's a really nice benefit of the NEALS program, which is in probably at 100 or so different academic medical centers. And we're, for our bank, we're actually sending for more advanced genetic changes, looking for advanced genetic changes to the New York Genome Center that is not clinically validated. So those are just for research. But there are lots of different clinically validated sites that you can send your tissues to. And not everybody wants to know, if they have familial. So, I, as a pathologist, if I'm doing an autopsy on a case, I will usually meet with families afterwards and do the very initial discussion about options for genetics if they hadn't done anything prior to death. But I always refer them to a genetic counselor because they're the ones who are trained to really handle the genetic questions. And some neurologists, have made that part of their practice and are really good at it. But I don't know too many pathologists that are. So I recommend you doing that. Let's keep going. What research progress would research progress more effectively by combining ALS and FTLD tissue findings and maintaining combined ALS FTD brain banks? Absolutely. And we're starting to do a number of different grant applications from our well-established brain bank through the Target ALS to try to expand that right now to include certainly FTLD, motor neuron disease, and other forms of FTLDs. The Alzheimer's disease centers often will also collect FTLD cases. I don't think they're so excited though about, and some are collecting straight ALS cases. So, I think we need to have because that's not what they're funded for by the U.S. government. However, there's so many overlaps. So there is an argument among our community that we should be including more diverse neurodegenerative diseases into brain banks and trying to go after more NIH funding for more holistic brain banks. Dr. Anthony says, how is ALS differentiated from post-polio? Is it possible that some of ALS cases with viral markers are patients who had mild polio in the past? It's a great question, Doug. When I was up at Dartmouth, I tried an experiment to vaccinate with the polio vaccine these poor rats, like overdose them to see if I could drive degeneration. Because there was some thought that maybe the reason that military people had a little bit higher percentage chance of getting ALS was they got more vaccinations than the rest of us. And we were never able to drive that kind of pathology. But it's an interesting, definitely an interesting thought. Could you speak more to relationship of TDP43 and C9orf72, since both are the most common findings? Do you feel that C9 drives TDP43 mislocalization in accumulation? I don't think so. I think they maybe are acting in different ways. The TDP43 has so many different interactions with different other proteins and DNA. And there are quite a few people that have TDP43 mislocalization and aggregates that have no abnormalities at all in C9orf72. So could there be a relationship for those people that do have those hexanucleotide repeats in C9orf72? Absolutely, there could be, and they could be affecting TDP43. But I think the opposite side of that with TDP43 doesn't always match up with C9orf72. I don't really know. The next is what's the relationship with retrotransposons? I really don't know, and I think
Brent,
that's
sorry. Let me explain. So, because you said that TDP43 loss can result in the elevation of retrotransposon, so that's
what Molly Hamill's group found. Exactly right, Mohammed.
So, if that's the case, can it be the same thing causing the integration of viral genome to the?
You know, it's possible. It's interesting. You know, TDP43, the very first papers that came out were people who were studying HIV. And then it was just a few years later that it was recognized amazingly in first frontotemporal lobar degeneration and then ALS. And so this relationship of this particular protein with the way our cells handle viruses and with neurodegeneration is really an interesting one that I think is ripe for a lot of research. We have time for, I think, a couple more. With SOD1 patients lacking TDP43 aggregates, I didn't mention that, but that's a great point to bring up. Do you think that SOD1 patients as a distinct subtype, or do you have insight into the clues that better unify ALS with TDP43 versus SOD1 mutation carriers? So I think that's also an excellent question. And I think that the abnormality at the cellular level, the biochemical level, maybe drives still motor neuron degeneration or glial atypia in both ways, but I think they are different. And what's interesting about SOD1, some of the mutations of folks that have SOD1 that run through their family drive the disease quicker, and actually some seem to be more protective, and people have a longer than that 2 to 5 year period kind of time point. Right now, I didn't talk about therapy very much, but if you have familial ALS, there are actually kind of some better chances that we may actually have some directed therapy. And so there are trials going on right now with using antisense against some of these abnormal SOD1s that are looking really, really promising. I'm not sure there are actually clinical trials right now against C9orf72, but there are certainly people thinking about that. So that's kind of where things sit there. Last one, I think we'll take, oh, nope. The last one was, please do your evaluation.
Right, no more questions. Thank you so much, Brent. That was an excellent lecture. Thank you for being also independent and taking care of the QA yourself. And also thank you for sharing that story with your dad. I think that was really, really nice to hear.
Great. Thanks, everybody. Take care.
All right, thank you again, everyone, for joining today's AANP Teaching Rounds presentation. We would ask that you take a few minutes to complete the evaluation that was entered into the chat box. Completion helps ensure accurate reporting to the accreditation board. It may not display as a link, so please copy and paste it into your web browser if needed. The PowerPoint slides and recording will be posted to the AANP website in the next week. Thank you again, Dr. Harris, for an excellent presentation, and this concludes our session for today.